and we're going to kick off today with the Clinician Scientist Award presentation. So I'm delighted to introduce Alexander Claussen. He was the recipient of the 2023-2025 grant, which the good news was for him was this was cut short because his project did receive NIH funding. But we did kick it off. And Dr. Claussen is an assistant professor at the University of Iowa. He completed his fellowship in neurotology at UCSD and a clinical research training program at NIDCD. Take it away.
Do you just okay, great. All right. Well, thank you for that introduction, and thank you to the AOS for kind of supporting some of this early work as a new investigator, new faculty. Today, I'm going to be talking about some of the work we're doing, looking at neo-ossification in the cochlea after cochlear implantation, both in humans and mice. I'm going to kind of broadly go over some of the experiments that were supported by this award and some of our preliminary data. I have no personal financial disclosures, but this work was supported by the AOS, now the NIH, and some support from Cochlear as well. So, post-CI cochlear neo-ossification, we know it's a common finding in a lot of human cadaveric temporal bone series. Why is this a problem? Why is it worth considering? Well, there's some evidence showing that maybe the degree of neo-ossification correlates with word recognition scores. There's some radiologic data showing that maybe it's associated with the delayed loss of residual acoustic hearing. We know from impedance studies in other parts of the body that bone generally has, worse impedance, higher resistivity than other tissues, which can impact implant function. And it really can be a barrier to other future CI technologies, including CI revision, optogenetic stimulation strategies, etc. And it's also an issue that we can see sometimes in other near-field neural prosthesis, not just in the ear. Heterotopic ossification can sometimes affect things like DBS stimulators as well. On the right, you're hearing showing some data from some other groups, some great work from Mass Ioneer and others showing this neo-ossification in the cochlea after cochlear implantation, both on histology and radiology. So at Iowa, a lot of the work we've done is looking at cochlear implantation in the mice and really studying cochlear implant biology. And we similarly see this robust neo-ossification in the mice. Obviously, it happens a lot sooner, a lot quicker, more robust in the mice, but it makes it a nice model to study it in. We can see this neo-ossification as early as a couple weeks afterwards and really progress after that. These are some histology and micro CT findings we've had from the lab showing some kind of interesting patterns of this neo-ossification. One of the benefits of the mouse model is we can really catch this process in the middle of it forming rather than years later when it's really been a mature response. And we see some interesting patterns. On the left here is a histology of the scala tympani showing what looks to be a case of maybe just direct endosteal extension of this neo-ossification right around here extending to the electrode. But one of the more common patterns that we see are these islands of really de novo mid-scala neo-ossification, where it's forming in the middle of the scala tympani, separate from the boundaries of the endosteum, which I thought was pretty interesting. We've done some studies to try and look like, is this a materials-related effect? Was there more bone forming around the electrode platinum-bearing surfaces versus the other? And there's maybe a trend toward that, but it wasn't very strong. And so all these observations, made me think there's probably some cochlear predisposition to ossification. We put implants at other sites of the body, and we don't see neo-ossification, when we put it under the skin or in the muscle. So it's not purely a materials foreign body related response. Obviously, we see it with other cochlear insults, meningitis. Yesterday, we were shown nice images showing it after a tumor. It made me think, what factors are there maybe contributing to this? One factor really implicated in heterotopic ossification, other sites of the body, is hypoxia. And I thought, well, this is all forming in the mid-scala, which is normally a relatively acellular place, potentially hypoxic. And is there a role for hypoxia really driving a neo-ossification process, which in that setting usually proceeds through endochondral ossification, not an endosteal extension? There's several other interesting aspects of the cochlea that make you think, is this going to be similar to heterotopic ossification at other sites in the body or different? The oda capsule is relatively senescent from baseline osteoprotegrin expression. It has slightly different derivations um, relative to the calvario and the appendicular skeleton being more paraxial mesoderm derived with some neural crest derivation compared to other locations. So might there be different osteoprogenitor populations as well? So really some open questions on CI biology, and really is neo-ossification actually having an impact on CI outcomes and really differentiated from fibrosis. And so in this study, we were trying to at least preliminarily look at those questions through two sets of experiments in humans and mice. First, we were looking to really characterize neo-ossification over time using photon counting CT and really correlating it with impedance measures and ECAP measures of neural health. Do these sites of neo-ossification correlate with neural dead zones? Are different impedance markers really reliable for detecting neo-ossification? I mean, really, what is the time course of it in humans? And then in the mice, really trying to identify what is the biology of neo-ossification, what is the fundamental process, what are the osteoprogenitor cells? And so I kind of go over some of this preliminary work, focusing a lot on point two here. I'm going to briefly go over some of our human studies that are still ongoing. I think we'll mature all these patients and get all the data by the end of this year. So I don't have all the final data to present, but I'll show what we're working on and what this award kind of preliminary helped us start doing. Um, Kind of grabbing patients from the DEX CI study. We've been able to grab 40 patients of people who've gotten either a DEX CI or a standard. And we're doing serial radiologic measurements with photon counting CT up to a year in a separate preliminary kind of proof of concept study. We showed what we think is able to detect bone formation in the cochlea with the photon counting CT. And so we're going to have that radiologic evidence of it. And then simultaneously performing repeated regular impedance and 4-point impedance measurements and ECAP derived measures to really assess if there's local bone formation electrode, can we detect a local neural dead zone around there? Can we detect this on impedance changes? And so that data is still pending, but we'll get some interesting results from that. I'm going to talk more about what we found out about neo-ossification and CI biology in the mouse using our previously published model of mouse cochlear implantation. So in the mouse, as I said, there's a really robust neo-ossification response that happens as early as 10 days. We start to see some bone formation. On the right here on some micro CT data, you can see on the upper right hand at 10 days post-implantation, there's red new bone forming in the round window area. We usually see it at some point in the base, and by 28 days and 56 days, it's really extended much more apically to about the distal point of the implant. On the left here, we've done a lot of hard staining to really assess what is the composition of this neo-ossification and see if we can detect things like, is there a cartilage intermediate to suggest this is endochondral ossification and not a direct endosteal extension? What we're seeing here is a Movats pentagram stain with the Alshan blue component, potentially staining a cartilage matrix green. And so we can see that by 10 and 28 days here. You can see that green staining area in the scala tympani, suggesting that this may be a cartilage matrix preceding the actual bone formation. And we have some preliminary histochemistry data showing this as well. These are really zoomed out pictures, so you'll have to take my word for it when we're looking at high mag on this. But some of the patterns we're seeing that correlate with this is early on high SOX9 expression in the scala tympani infiltrate, which is suggestive of an initial chondrogenic differentiation or fate at 10 days. And then by 28 days, we see the SOX9 expression decreasing these areas of bone formation and taken up by RUNX2 and osteocalcinin expression, which is indicative of osteogenic differentiation. So together, this shows that at least partly it looks like there appears to be some endochondral neo-ossification process happening and not just all a direct endosteal extension. Concurrent with endochondral ossification is typically a hypoxic-driven response, and we see evidence of that as well. On the left here, we're seeing nuclear and perinuclear transition of HIF1 alpha staining and a lot of the cellular infiltrate in the scala tympani, suggesting there is a hypoxic process. And corollary to that, we see a large presence of neoangiogenesis starting as early as 10 days and proceeding onward after that, which we're seeing on the right-hand side with a CD31 GFP reporter. So there's a lot of parallel conditions happening, including a hypoxia-driven response that could support a process of endochondral ossification being responsible for this. And so that's really what we've seen is some preliminary evidence suggesting that there is an endochondral ossification process to neo-ossification in the cochlea. We're doing ongoing multiplexed IHC experiments to confirm the identities of this chondrogenic and osteogenic cells, and doing a transcriptomics and lineage tracing experiments to really identify what are the osteoprogenital populations, either in the cochlea or are they migrating into the cochlea after injury. All right. I just want to thank everyone who's been part of this work from multiple institutions, and really thank you to the AOS for kind of jumpstarting this line of inquiry. Oh, yeah.
We have time for questions.
Unsurprisingly, that's totally cool.
Yeah.
Photon counting CT can actually do some chemical analysis of tissues. Have you looked at all into what might be needed to modify the CT scanner so as to differentiate, say, cartilage from bone, and then you could watch this process over time?
We've done some preliminary experience in cadaveric temporal bones, essentially putting bone in the cochlea to see if we, can confirm we're seeing neo-ossification, but I will have to talk to you more after that, more about that afterwards.
Um, Beautiful work, and I love that you're looking at it also molecularly. Do you have any thoughts about how to prevent this process and possibly counter key regulators of this ossification or neoangiogenesis?
Yeah, the you know the concept one part of this line of investigation is seeing if this process is similar to heterotopic ossification in other parts of the body, because there are modalities to treat that. Some are not so good for the cochlea, which is radiation, which people commonly use. But there's things like retinoic acid antagonists that people use to treat this and um, other pharmacologic modalities. And so one goal would be to see if there's similar factors driving this and then use similar treatment strategies used in other parts of the body.
Really nice talk and data. In your single cell approaches, are you planning on looking at maybe other mouse models where you have heterotopic ossification in other places so you can have a comparison to what's going on in the cochlea?
We have thought about that. We've looked into some kind of tendinopathy models. It's difficult because in the ear, again, it has different embryologic derivations, and so in other areas of heterotopic ossification, there's resonant mesenchymal stem cells that they've identified responsible for that have lateral plate mesoderm derivations. Whereas in the cochlea, if that's the same process, it would probably be from paraxial mesoderm. It may have different factors, but that is one thing we've considered.
Thank you very much.
Yep. Thank you.